Yeah,
Okay.
thank you very much.
Thank you. Okay. The next speaker is Dr. Shangwei Zhong from Beijing University, China. He will give us a talk about the role of 5B in light and temperature perceptions. Dr. Zhong, yeah, please.
Yeah, thank you. Thank you for the introduction, and I also want to thank the committee for give this opportunity. It is my great pleasure to have the chance to introduce the studies in my lab. The title of my report today is Light and Temperature Perception by Fatcom B. The sun provides us light and heat at the same time, which are essential for plant growth and also for the human beings. Also, light and temperature are usually correlated, but these signals are distinct physical factors. The signals of light temperature are essential for plants to determine the day of the time of day, season changes, and the positions. So one of the most fascinating questions in plant biology is that how do plants sense light and temperature? During the past decades, it has been identified several photoreceptors in plants. Among them, Fatcom B is a main red light photoreceptor. Fatcom B is synthesized as a PF form in the dark. When exposed to red light, the PF form will transfer to the PFR form, which is the active form, and the PF form of 5B will translocate into the nucleus, where it will regulate the expression of thousands of genes to produce the physiological responses. And this PF form 5B can be reversed to the PFR form by red light. So with this PR and PF form conformational changes, 5B can sense the light changes. In 2016, two science papers also found that 5B is functioning as a temperature sensor. When in Y-type, when the seedlings are moved to a higher temperature, the hypocoagulation will be promoted. In 5B mutant, the hypocoagulation is constitutively negated and did not respond to warm temperature. When you transfer the white of 5B into the 5B mutant, the white of 5B can restore these defects of 5B mutant and respond to temperature similar to that of white type. However, when transform this YB form of 5B, this is a PFR form locked 5B. These transgenic plants still did not respond to temperature. Therefore, these studies propose that the warm temperature will promote this PR form to PR form reversion that enables the 5B to sense temperature changes. However, besides of temperature, other signals such as shade avoidance, far light, and darkness, all of these signals will regulate this process. So how do plants distinguish these signals is still unclear. In our study, we expressed the YB in the mammalian cells, and we found that in low temperature 5B, YB will form food bodies, which is similar to that of in plants. However, when the temperature increased, these food bodies will disappear. disappear. And when the cells return to the low temperature, the food body will reappear. So this YHB food body formation can be reversibly respond to temperature changes. We also examined the recognition in plants and found that YHB food body did not respond to that. However, with the temperature increased, the food body of YHB will be reduced. So there are some questions about this. First, how temperature regulates the YHB food body formation? Because YHB is a PFR from knocked Fab, its protein activity cannot be regulated. So how temperature can regulate this food body formation? Also, we still don't know whether this YHB food body formation has a physiological relevance. Luckily, during the recent years, a new concept of nickel-nickel phase separation has been introduced to the biological researches, and especially for research study the formation of membranous compartments. Many bodies and stress granules are found to be formed through this process. So we wondered whether Fab for the body is formed through nickel-nickel phase separation. We examined the properties of Fab for the bodies in plants and found it exhibits the classical liquid properties in plants. We also expressed Fab in human cells, and we found also this Fab for the bodies also have the liquid properties. So Fab is likely undergoes nickel-nickel phase separation to form these droplets. Then we want to know how this separation is regulated. We analyzed the amino acids of Fab protein and found the N-terminal extension region of Fab is highly disordered. And this disordered region is probably regulate this phase separation. So we express the fullness Fab and the truncated Fab without the NTE, and found with deletion of NTE, the droplet formation of Fab will be totally abolished. When adding the different ADRs, such as ADRs FL2 from plants and FOS from animals, this ADRs can restore this food body formation. Therefore, this results indicate that the NTE of Fab is a phase separating ADR that promote the Fab, the droplet formation. We also examined the contributions of other domains of Fab in regulating this droplet formation, and found that if divided Fab with the N-terminal, C-terminal, we found the N-terminal unknown cannot form the food body. However, the C-terminal will continue to form the food body and did not respond to light. Fab C-terminal was previously reported to be a self-association domain. So we fused the Fab N with a gas tag. Gas protein is also a self-association protein. So we found that the gas fusion will restore this food body formation. We further examined the functions in plants. We found that the N-terminal Fab unknown that cannot form the food body have a partial function of Fab. However, when the Fab N was fused with gas that restores the food body formation capacity, the function will be greatly increased. Therefore, this genetic results indicate that the droplet formation of Fab can improve the physiological function. However, the mechanism is still unknown. By review this mechanism for Fab food body formation, we then investigate how temperature regulate this drop information. We still use the YHB to exclude the light effects. We found the temperature will reversibly regulate the YHB for the body. However, when the NT region is deleted, this truncated YHB can form for the body, but cannot respond to temperature. 5B system also form for the body, but did not respond to temperature. Therefore, the NT region of 5B sense the temperature changes to moderate the droplet formation of YHB. Then we want to know what this regulation for the body formation have a physiological function. In previous studies, have shown that transfer YHB into 5 mutant, the transgenic plants still do not respond to temperature. However, in our studies, surprisingly, we find in our transgenic plants, we found that YHB transform into 5 mutant can fully restore this defects of 5B mutant in response to temperature and display a very similar response like Y type. We further examined our transgenic lines, and we also found some lines like this have the similar phenotype like reported. So what's the reason for this different responses of transgenic plants in response to temperature? So we examined the protein levels and find that in these lines, the protein level of 5B is very high overexpression. So the effects of temperature on YHB could be attenuated. So when the YHB protein level is comparable as the Y type, these transgenic plants can respond to temperature normally. Because the YHB is a PFR locked form, so temperature could not recognize the protein activity of this protein. So how this droplet formation can recognize the function, the physiological function of YHB? We further examined the formation of 5B droplets. When you co-express 5B with interacting transcript factors such as PF3 and PF4, we found these transcript factors are recruited by 5B into their bodies. So in this 5B for the bodies, not only 5B protein itself, but also other components in the signal transduction pathway, such as the transcript factors, also will be recruited. So to constitute a high level of protein concentration in this droplets to accelerate the signal transduction. So in summary, in this study, we found that light is sensed by 5B through the conformation changes to regulate the protein activity of 5B. But temperature is sensed by 5B through the phase variation to regulate the 5B signal transduction efficiency. So through these two mechanisms, 5B can sense light and temperature at the same time, but also 5B can distinguish light and temperature. With the case of studies of 5B, now we have known that 5B is accumulated in the cytoplasm of dark growth citizens and in the dark growth citizens, and we are rapidly translocates to the nucleus in light. In the nucleus, 5B will interact with transcript factors such as PIFs to regulate gene expression. In previous study, the study have found that if 5B is detained in the cytoplasm, such as this, the physiological function of 5B will be completely abolished. So the cytoplasm to nucleus translocation is a rate limiting step for 5B for the transductions. However, even with extensive studies of 5B signaling transduction, we have many knowledge about this pathway. This is the rate limiting step of 5B new contraindication. The mechanism, the regulation also did not unclear. So in our studies, we found that when exposed the dark growth seedlings into red light explosion, we found that the red light will stimulate a very rapid increase of cytosolic calcium in the seedlings in less than 1 minute. And this cytosolic calcium increase will return to the basal level also very fast in 1 minute. So red light explosion can induce calcium signal in eternity seedlings. So we want to know what this calcium signal is functional relevant. So we use chemical treatments to repress this calcium increase. And we found that with chemical treatments, light regulated gene expression will be repressed. More importantly, we found that the light-induced 5B new translocation will also be repressed. Therefore, this calcium increase is required for the light-induced 5B new translocation. This is a very exciting finding. So we then want to know how this calcium signaling can regulate the 5B new translocation. By analyzing the calcium binding potence with 5B, the interaction, we found two calcium-dependent protein kinases, CPK6 and 12, can interact with 5B. By in vivo QIP, we show that when the seedlings transfer to redundant elevation, the interaction between CPK and 5B were greatly increased. We further examined the interaction by using in vitro pull down. We found the interaction of 5B and CPKs are very specific. When 5B is activated by light, but without calcium active CPK, or when it's a calcium active CPK, but the 5B is not activated by red light, none of this combination can produce strong interaction. The strength is very weak. However, only when the 5B is photoactivated by the red light and the CPK are activated by calcium at the same time, then the interaction will be very strong. So the interaction is in a manner of dual dependence of light and calcium. We also found that when the CPK6 and 12 are deleted, mutated, the light-induced 5B nuclear location will be largely abolished. Also, this signature pathway also be repressed, like PIF degradation or gene expression. So CPK6 and 12 are required for 5B, the nuclear import and the signaling pathway. Because the CPKs are kinases, so we want to know whether the light can regulate 5B phosphorylation. We found that two sites, 080 and 0106, the two sites, the phosphorylation level was significantly elevated by red light. So we then examine whether CPK can regulate, can phosphorylate 5B at this sites. By this in vitro phosphorylation assays, we found that when the 5B is activated by red light and the CPK activated by calcium, the CPK will phosphorylate 5B, and this depends on these two sites. So these two sites are phosphorylated by CPK during the doctor transition. Whether these two sites are regulated or involved in 5B's nuclear translocation. So we use the transgenic analysis to find that when the doxorubicin is exposed to red light, the 5B, vital 5B protein will be rapidly translocated into the nucleus. However, the 2A with the two sides phosphorylation defective protein, the translocation will be abolished. So the two-size phosphorylation is indispensable for that induced 5B nuclear import. We further investigate the physiological function of this regulation. When the seedling is exposed to light, the candidates of Y-type will be rapidly opened and expanded. However, in the mutation of 6 and 12, the candidate still closed and unexpanded, similar to that of 5B mutant. Also, when you transfer vital 5B into the 5B mutant, the defects will be fully restored. However, when you transfer the 2A form of 5B into 5B mutant, the still maintain the 5B mutant phenotype. Therefore, we show that the CPK6 and 12 through this two-size phosphorylation regulate 5B nuclear import is essential for the deterioration transition of 5B. In summary, in this study, we've shown that when seedlings emerged out of the soil, the red light will stimulate an acute sonic cache increase to active CPKs. This active CPK will directly interact and phosphorate the 5B to control the 5B nuclear translocation. So this mechanism, the seedlings can be rapidly undergo the detonation transition when it emerged out of the soil. And lastly, I want to thank the students and posters working in this project. And I also want to thank the collaborators, our founders, for supporting us researchers. Thank you very much. And any questions are appreciated.
Okay, thank you. Thank you, Zhong. Very interesting findings. So is there any question from the committee members, please?
Hi.
Hi.
Hi, Shangwei. Nice talk. You talk about the 5B can serve as both a light sensor and temperature sensor, right?
Yeah.
In
Yes.
nature, we know that light and warming come together, right? When sun come out, it provide light signal and also rising temperature, warming temperature, right?
Yes. Yes.
But in your essay, warming temperature is promoting elongation of the hypocaudal, but light inhibit hypocaudal growth. Is that right?
Yes.
So how do you explain this, light and temperature antagonism? Maybe balancing the growth speed. Have you thought about that?
Yeah. In the when the light active the 5B, the 5B will repress the hypernegation. So light will repress hypernegation, and warm temperature will repress the phosphorylation of 5B. So repress the 5B signal transduction efficiency. So so the warm temperature will repress 5B function, so will promote the negation of hypocaudal. So this phenotypes can be explained by this mechanism. For in the nature, when the temperature is higher, is in warm temperature, the plants will try to escape from the hot in the surface, soil surface high temperature, so will grow very fast to protect themselves. So they can repress the light function to promote negation. So it's a kind of adaptation strategy, adaptive strategy for plants used to in this high temperature stress environment.
Thank you.
Yeah, thank you.
Yeah, maybe also a question. Nice talk. So everything starts with the calcium increase in your last story. So so the red light is inducing the calcium increase. I guess this is phytochrome B dependent. This calcium increase.
Yeah, it is.
And secondly, where the calcium comes from? Is it from the apoplast, or is it released from the ER, or what kind of? Is it known which kind of calcium channels are involved for this calcium increase in the cytoplasm?
Yeah, that's a very good question. We found that this RNet-induced calcium increase is dependent on FAB, because with the FAB mutation, the RNet cannot promote this stimulate this calcium increase. So it's dependent on FAB. But phytochrome B is not a air channel, so it's not itself mediate this calcium increase. So it proposed possibly FAB can interact with some air channels, or directly or indirectly can regulate this channel's activity, so regulates the calcium increase. This calcium is from the outside of the cell, it's not from the inner cell. Yeah. Okay. So the channel haven't identified which channel is, but the specificity is controlled by the FAB itself.
But how do you know it comes from the apoplast? Because I think that might also not be easy to demonstrate whether it comes from outside of the cell or from the ER. Because in many cases you have a calcium release from the ER, but how do you know this difference?
Yeah. We use a chemical that can block the plasma air channel that will okay, this the calcium input. Yeah.
Okay. Thank you.
Thank you.
Well, if there's no questions, thank you for your next talk, Dr. Zhong.
Thank you.
Okay, let's move to the last talk.